In this section, I'm going to talk about specimen preparation. Typically, when we want to look at something under a microscope, there's some prep that has to happen. The method of specimen preparation depends on the condition of the specimen, whether it's living or preserved, and the aims of the observer. Do you want to observe it, identify it, look for movement? The type of microscopy available is also important. If it's a dark field versus a bright field versus a fluorescent microscope, different prep is going to go into play. Wet mounts or hanging drop mounts. So in wet mounts or hanging drop mounts, cells are suspended in a fluid that allows the organism to stay alive and move. It provides a true assessment of size, shape, arrangement, color, and motility or movement. In a wet mount, usually you put a drop of water directly on the slide and then put a cover slip on top of it. Advantages are it's quick and easy. Disadvantage is that it can damage larger cells and it's susceptible to drying. If you let a wet mount sit out on the counter for too long, it will dry up. It can also contaminate the handler's fingers. So even though we wear gloves in the laboratory, if you are directly putting that cover slip on top of the slide, it can contaminate the handlers. In a hanging drop mount, it's prepared with a concave slide, a Vaseline adhesive or sealant, and a cover slip. It overcomes the disadvantages of wet mounts. So basically what you do is you put a drop on the cover slip, and you have the cover slip surrounded with some type of Vaseline or adhesive or sealant, and then you put it on top of the depression slide so that the drop hangs down into the depression slide and the Vaseline forms a seal to prevent the drying of the specimen. The smear technique, you spread a thin film made from a liquid suspension of cells on a slide and then allow the slide to air dry. This is common for making blood smears as well. So we put a drop of blood on the slide and then we can smear it and observe that way. You may be wondering why I'm talking about blood when we're in a microbiology course. Well, there's a lot of microorganisms that you can identify in blood specimens. One of the biggest ones being malaria. So fixing smears, you can heat fix them. Heat fixing is when you apply an amount of heat directly to the growth or the microorganism that's on the slide. And it kills the cells and secures them to the slide and preserves the natural components or the components in their natural state within the cell. Alcohol and formalin can also be used for fixation, and those have their pros and cons. Alcohol usually dehydrates, formalin can be toxic, so heat fixing is a really safe and easy way to just kill everything on the slide and fix it exactly where it is. Staining smears. So it's very difficult to see microorganisms without stain. Staining provides contrast, and we talked about contrast previously. If we have stain, it allows you to differentiate between a cell and a non-cell in the background. It makes inconspicuous features stand out. Dyes impart the colors to the cells by becoming affixed to them through a chemical reaction. So there's basic dyes which have a positive charge, and there's acidic dyes which have a negative charge. And using those different dyes allows us to use the different staining procedures. A positive stain uses a positively charged stain. The surface of a typical cell is negatively charged, and if you've ever heard of the phrase opposites attract, using a positive stain allows the stain to be attracted to the surface, the negative surface of the cell. This typically sticks to it permanently and it kills the cell. In a negative stain, the stain is negatively charged. So if you have negative charged stain next to a negatively charged membrane, they're going to repel each other. So negative stain usually just stains the background, allowing the specimen to remain untouched and alive. So this is a picture of a positive stain. You can see that the stain is actually stuck to the surface of the cell. And here's negative staining. You can see that the stain is in the background and the surface of the cell is clear. Negative and positive stains don't have anything to do with gram staining. So we're going to talk about gram staining, and that is a major part of being a microbiologist, is learning about the gram stain and its importance. But positive and negative stain is just talking about the stain itself. So a positive stain includes crystal violet, methylene blue, saffron, malachite green. Negative stain includes nigrosin or India ink. When we talk about the gram stain, you'll see that the gram stain procedure uses all positive stains. It uses the crystal violet and the saffron. A simple stain uses one color. All cells are stained one color, and all structures have the same color. Differential stains allow you to see different structures because different structures will stain different colors. Examples include the gram stain. So the gram stain is a differential stain in which gram-positive bacteria stain purple and gram-negative bacteria stain pink. Again, gram-positive and gram-negative do not have to do with the positive or negative stain. It has to do with the structures within the membrane or the cell wall of the bacterium. And we'll talk more about this once we start talking about bacteria in this course. An acid-fast stain differentiates acid-fast bacteria from non-acid-fast bacteria. Acid-fast bacteria stain pink, non-acid-fast bacteria stain blue. What is the importance of being acid-fast? Acid-fast bacteria have mycolic acid in their cell wall. And I'll write that term down. Mycolic acid is a wax-like substance. If you've ever put water on a candle, on a wax candle, you know that it beads off and it falls away. That's kind of like what happens when you put an antibiotic near the surface of an acid-fast positive bacterium. Mycolic acid will prevent the penetration of that antibiotic into the cell. This is a major problem because Mycobacterium tuberculosis, the causative agent of tuberculosis, and Mycobacterium leprae, one of the causative agents of leprosy, both are acid-fast positive. So these are really difficult to treat with antibiotics, and they typically require a 9-month plus course of antibiotics. The endospore stain. So endospore stain is used to distinguish endospores from vegetative cells. Endospores are really tough, durable, dormant forms of the bacterium. So sometimes bacteria produce these, and only certain species can produce these, but they will produce them in response to stress. If you've ever heard of C. diff, Clostridioides difficile, that causes diarrhea and usually pretty prevalent in hospitalized patients. C. diff can start to overgrow when a patient undergoes a course of antibiotics. That antibiotic treatment kills away their natural bacteria, predisposing them to overgrowth of the bad bacteria, and C. diff being one of them. C. diff is an endospore former. So when this person is having this diarrheal issue, those spores get into the environment, and then anyone that enters into the room can come in contact with the spores. And as soon as the spores enter into a new host, they will hatch open and turn into the real deal vegetative bacterial cell. They have the potential to spread in hospitals like crazy and spread in long-term care facilities and so forth, other types of clinics. So these are very dangerous. What makes them more dangerous is that they are resistant to most forms of disinfection. So only sterilization methods can get rid of endospores. They resist UV light, heat, all sorts of stuff. So they're really difficult to get rid of, and that's why we care about endospores. In order to diagnose certain bacterial infections, we want to know if that microorganism is an endospore former. That's one of the distinguishing factors that allows us to say it's this bacterium versus this bacterium. So we have the staining procedure that will specifically stain spores. This staining procedure, just like the acid-fast staining procedure, they're both pretty dangerous. Dangerous meaning that there's a steaming method or a steaming procedure in there that requires you to steam the cells, and steaming causes the evaporation and aerosolization of the stain, which can be harmful and can cause some pretty nasty reactions. So you have to do it in a fume hood, and it's tricky to perform that way, so it's safer to just not perform the experiment at this level, of course. So with the endospore stain, it detects endospore-forming members of the genre Bacillus and Clostridium. Here you can see the endospores are in pink, and the vegetative or living, breathing, moving cells are here in purple. Sometimes or in blue. Sometimes they will put malachite green instead of the pink, and the spores will be green in color. Special stains, some staining techniques are differential and are also in the special category because they pinpoint particular characteristic. So the capsule stain stains the capsule. In this staining procedure, the bacterium is stained and the background is stained, leaving the capsule glowing white around the organism. A capsule stain is used to identify pathogens such as Cryptococcus. Cryptococcus neoformans is a really bad cause of meningitis. It's a fungal, it's a yeast specimen. So if this gets into the CSF, it can cause some pretty dangerous effects, including death. So Cryptococcus neoformans, if we want to investigate a cerebrospinal fluid sample to determine if it's positive for this, we will do a capsular stain. Flagellar staining. So flagellar staining procedure stains the flagella. The flagella are really skinny and they move pretty quick. So in order to stain them, we have to put a thick stain on them. It's kind of like coating them with the makeup mascara. So it's like putting this thick coating on them, which slows them down and makes them appear thicker so that they can be visualized. We care about this because certain bacteria have different flagellar arrangements, and their arrangements can give us clues to their identity.